Thank you. Everyone, mic is good? Yes? All right. All right, thank you so much. It's such a pleasure to be here today and give this week's Beck talk. So I study life history trade-offs across different biological organizations. For this talk, I'm going to focus mostly on organismal evolution and how organismal evolution and life history trade-offs in cancer might give us some insight into studying human variability in the disease. Okay? First, I'm going to start off with some brief statistics to get everyone up to speed on cancer rates in the U.S. This is 1 in 3 women in their lifetime will develop cancer, and 1 in 2 men in the United States. The risk of dying from cancer, 1 in 5 women have a lifetime risk of dying from cancer, and 1 in 4 men in the U.S. have a lifetime risk of dying for cancer. And this means that probably cancer impacts almost every person in the U.S., either through their social network or personally. I'm just going to give you a brief look into what cancer is at the cellular level. If anyone has any questions for clarification, please just stop me. I just want to get everyone on the same page. So we are multicellular beings, and our cells divide, okay? Every time a cell divides, there's a probability that there could be a mutation in the DNA, okay? So here's a cell with this little star indicates a DNA mutation, okay? And then this cell typically stops their cell cycle and has to make a decision process right here. So it can repair the mutation. Typically when a cell repairs its mutation, it's not perfect, okay? So there's always risk to going down this path. It could lead to a point mutation that stays within that cell forever. Cell could have mutation, assess, and decide it's going to kill itself, something called controlled cell death, also known as apoptosis, okay? Or the cell cannot repair or kill itself, acquire the mutation, and keep dividing, okay? And this can happen time and time again. The cell has these acquired mutations, and throughout the course of an individual's lifetime, or the cell lineage lifetime, it can accumulate mutations. Some of these mutations might land in important genes that influence the proliferation of the cell, and at some point the cell can lose constraints into what is normal for a multicellular being. Abnormal proliferation occurs, and then this is the consequence you get with cancer, okay? And I'll go back to this point, but all else being equal, we need to think when we get mutations, it's important for a multicellular being, the number of cells that are dividing, we think of it as a numbers game. All organisms being equal, if you have more cells, there's a higher probability that you can get a mutation that could turn into cancer. But how long you live, you can have a greater accumulation of mutations and a probability of getting cancer. And I'll come back to these 2 points of the number of cells and how long an individual lives. Okay, so if we go back to this, I don't know, how many people are shocked to hear these statistics? A little bit. When I first started studying cancer 5 years ago, I was slightly shocked. I didn't really know that we had, at least women have, a 1 in 3 lifetime risk of developing cancer. And I really wanted to know if this was a uniquely human trait, or if we're at an increased risk living in the Western world, and what's really going on with this. And so I thought the first place I'd start is looking at it from a phylogenetic perspective. How uniquely does this cancer risk land in the human population versus other animals? If you scan out on the tree of life, and if anyone has a pet, they might know that humans are not the only ones that get tumors. Tumors occur very frequently in domesticated species, like our loving pets. But if you keep expanding out, how far back can you go? How far back do you see cancer arising across the tree of life? when did cancer arise in this tree of life? Okay? And I would argue that it's gone back to the evolution of multicellularity. Okay? A single cell essentially cannot get cancer. It can get mutations that affect its fitness. But it's only multicellular beings that can actually get cancer. And actually, complex multicellularity has evolved independently multiple times across the tree of life. And by exploring how multicellular organisms have not only evolved cancer suppression mechanisms to become bigger and more complex, differently across these different independent lineages of the evolution of multicellularity is a really important point in figuring out novel cancer suppression mechanisms across the tree of life. Okay. So we had this idea, a few colleagues of mine, to look across the literature and see essentially if cancer is a problem of multicellularity. Can we really map it on how often do we see cancer across the tree of life? I apologize, this figure is really tiny and hard to see, but what we did here is if you see the red boxes, that was a cancer-like reported in the lineage studied. A yellow box was a cancer-like phenomena, and green was no cancer reported. This was a literature review. That doesn't mean the organism didn't get cancer, it's just it's not been reported as of the study in 2015. The black, gray, and white indicate either complex multicellularity, simple, or unicellular. And what we see is we do see cancer red or yellow occurring across the tree of life in different independent mechanisms of multicellularity. There's a few organisms here, like these sponges, where we see no reports of cancer given. We have some collaborators right now researching this in more detail, trying to actually irradiate these sponges and see if they form tumors. So far, they have not been successful, so that's a little bit exciting of a report that can come out of just a survey of looking this, and then we can start looking at the mechanisms of why. Okay? And so coming from a different framework and thinking about the origins of multicellularity and in cancer, colleagues and I like to think of it as, so we have these foundations of multicellularity, all right? So this is somatic cooperation. A cell has to get rid of its cellular fitness, suppress it for organismal fitness. And there's basically 5 foundations we came up with. That you have to have proliferation inhibition. You check yourself and also your neighbors to not proliferate uncontrollably. There's controlled cell death. That is, if you do get a mutation, you have this apoptosis event that happens. There's allocation of resources across different cells. Division of labor. We have specialized cells that do specialized functions. And you all have a shared environment or neighborhood. So this is what we consider somatic cooperation. And essentially what cancer is, thinking about it from this framework, is that it's somatic cheating. It's a breakdown of cooperation across multicellular organisms. And so suddenly you have uncontrolled proliferation, inappropriate cell survival, resource manipulation. Tumors need resources. They create markers that recruit blood vessels to get new resources into the tumor. There's dysregulation. They suddenly don't care what kind of tissue they came from. And they degrade their multicellular environment, throwing a bunch of waste products into it that only then the tumor can survive in. Okay. So cancer is this evolutionary ancient disease, and I would argue that it is a consequence of multicellularity. Here's just a few examples that we reviewed in our 2015 paper looking at cancer across the tree of life. This is one of our favorites. It's a crested cactus. It's actually in the botanical gardens it's in Tempe, near Arizona State University. And it's thought that these fasciations, this fan, so the cactus is supposed to be straight and it's got this fan phenotype, is due to somatic mutations in it. And botanists love finding crested cactus for their gardens. Here's an example of an abnormal proliferation on a fungi. This smoothing of the coral is loss of differentiation. So we can see these different characteristics all across the tree of life. Although one thing we did find in our survey is that there's variation across the tree in life and what organisms are better at suppressing cancer than other organisms. And that leads us to ask the question, why are some species better at suppressing cancer than others? If cancer is an evolutionary agent, why haven't all species gotten really good at cancer defense mechanisms? And the answer is likely a few different explanations. That there's trade-offs, that we're potentially a mismatch in our environment, and a term of antagonistic pleiotropy. I'm just going to briefly give examples of each of these. So antagonistic pleiotropy, so these benefits early in life can lead to poor outcomes later on in life. In the context of cancer, we can think of it as a selection for growth. So having more cells, bigger organisms generally are less susceptible to predators, have greater reproductive success than their smaller rivals. But as I told you earlier, cancer is a disease and a probability of the number of cells. So having more cells increases your risk for having a mutation that could lead to cancer. But also growing fast, rapid cell division. You're unlikely to be able to be perfect in repairing your DNA if you have to grow big fast. And you could see how being big or growing fast could be adaptive in certain environments, but could lead to cancer consequences later on in life. I give you the example of melanoma in a male platyfish. This black spot on this fish tail is actually melanoma. And these fish, the females select to mate with the largest male. This is a growth factor mutation, and these males are typically larger. And so the females preferentially mate with the males that have this mutation, but they go on to get melanoma. So this mutation stays in this population of fish. Here's another, sorry, you guys are eating lunch. I should have warned you. This is the only gross picture. So these are antleromas in free-ranging deer. This is a male deer, and it's common enough to have a term called antleroma. We don't know much about it. It's mostly observed in hunters, but it's really interesting because the antlers that male deer grow are the fastest growing mammalian appendage. They grow them yearly and then they shed them. You can see how both of these, the size, but also the extreme ornaments on an organism would be beneficial for that organism's reproductive success, but could be at a cost to increase risk for cancer later on in life. Okay? And this is something that you can think of as trade-offs between reproductive success and cancer defense mechanisms. The other example of potentially why we might see cancer more often is through evolutionary mismatch. So we're just not adapted to the modern environment. A term that we should all be pretty familiar with here in this room. One example I wanted to give was of the domestic hen. And ovarian cancer is a big problem in domesticated hens. They've been bred to lay eggs daily, and they're the only, so far, which we'll see, I have more data now, but so far they're the only non-mammal or non-human ovarian cancer cases across species that we've found so far, okay? So this is just an example of mismatch. And the third thing I wanted to highlight is life history trade-offs. And some of you might be a little confused on how there could be trade-offs in cancer defenses. But I would argue that cancer defenses are a major component of somatic maintenance. And that includes DNA repair, cell cycle control, deciding whether to repair the DNA or go on to apoptosis. And also importantly and very costly is immune function. Our immune system can detect cancer cells, is detecting mutated cells all the time and clearing them from our body, okay? Usually through T cells. Okay, and so from this prediction, we can maybe say there's a relationship between cancer rates and life history strategies across multicellular organisms. And organisms that live in harsh environments with unpredictable resources invest in reproduction over survival. Where organisms that live in predictable environments and low predation invest in somatic maintenance over reproduction. And we can also map this onto maybe looking at somatic maintenance as cancer defense mechanisms. So from a life history standpoint, we would predict that mice would have higher rates of cancer than elephants, long-lived, large-bodied elephants, okay? And this is fascinating because I was formally trained in life history theory, but when I started studying cancer, there's something that I read about and I thought it was beautiful. It was called Peto's paradox. So Sir Peto, many years ago, made this observation. He said, humans are much smaller than elephants. Elephants have many more cells than humans. All else being equal, if we have the same cancer defense mechanisms, we should expect elephants to have much more cancer than humans, okay? And humans to have much more cancer than a small mouse. They have less cells. Elephants don't have bigger cells, they just have more of them, okay? All else being equal, they have a higher probability of getting that mutation that could lead to cancer. But this isn't what we observe, okay? We do not observe high rates of cancer in elephants. He didn't have much data to show this, but he knew elephants weren't dropping dead of cancer all the time. And so he hypothesized that there must be extra cancer defense mechanisms bringing down this rate of cancer in elephants, okay? Okay, and so does the data match the theory? So far I've given you a bunch of framework leading up to life history theory. And when I first started studying cancer a few years ago, we did not know the cancer rates in elephants. We didn't even know cancer rates in our closest living relatives. We knew that chimpanzees and other primates did get cancer sometimes, but we didn't know the rates and we didn't know what types. And this was published in 2015, and it talked about the rarity of epithelial cancers in captive chimpanzees, although we didn't have much quantitative data. And I'll come back to this because it seems like with newer data, this is, seems to be a shifting story. So previous work looked at a book published of animal deaths in the San Diego Zoo, and we had a grad student go through and pull all the cancer data. It's just a big book of what the animals died of. Really fascinating literature. I actually think it is fascinating literature, but there's 36 mammals in this study, and we're just looking at, so they did necropsies, and if there was a tumor found in the necropsy, that got marked into a category out of as many animals that were looked at in necropsies, and you got a % of neoplasia across all the animals, okay? And here's it. This is the first empirical evidence to support Peto's paradox. So again, we're looking at necropsies with tumors. And then we're looking at body mass times lifespan, okay? And what we see is that there is no relationship between body mass, lifespan, and cancer rates in these 36 mammals, okay? I would like to point out this is the Tasmanian devil up here. This is not, some of you might know that Tasmanian devils get a contagious cancer. It's a facial tumor. These animals did not have facial tumors. It was a zoo population. They were immune from it. So even before they started getting facial tumors, they still were getting high rates of cancer. Does anyone know about the contagious cancers in other animals? We've now discovered 5 different species that have contagious cancers. We can talk about it in the discussion. It's quite fascinating. It's usually organisms that have gone through a genetic bottleneck, but one of the cases is in dogs. Okay, and so understanding the evolutionary and environmental pressures that in animals that can enhance cancer defense mechanisms can really enhance what we know about humans. And so I really wanted to gather more information like I showed you before. We're really excited. We got data on 36 animals. This paper came out. Everyone was very excited. And I thought, gee, we need more animals. So I started calling around to a bunch of zoos and saying, hey, I'm doing a study. I would like to know if we can get some of your necropsy records. Do you ever report when an animal gets cancer or not? And the zoo said, of course, yeah, that's great. And so you fill out a bunch of forms, and they're like, what species are you interested in? And then I said, every animal that has ever existed at your zoo, and then they never called me again. So it has been very challenging getting this data because it's there, it's just people are overworked, and it's a lot of paperwork, and a lot of it's not electronic. Fortunately, I've met 2 amazing people that are in the vet community, and if you're trying to merge fields, you should probably get allies within that community, and that really gets your foot forward. And the amazing thing is I was approaching it so wrong. I was like, I'm interested in human cancer. Give me your cancer on or give me your data on animal cancer. And that's really not the approach to do. Lesson learned. We should look at it as a One Health perspective. These animals are getting cancer. They're family pets. They get chemotherapy. They're considered patients, and these vets care deeply about getting better treatment for these animals and also understanding the risk factors within these animals as well. So we've joined teams. I'm very happy. I think they're happy too. And now we're finally starting to get some data, and I'm really excited about this. So previously, we just had the San Diego Zoo data. It was only 850 species, or 850 individuals, 36 species. We now have data from the Northwest Zoo pathology. That's over 114,000 individuals, 13,000 species. Also an exotic pathology service, adding another 550 species. And then some wonderful data from the Duke Lemur Center, really bumping up our sample size. We're still very much in the process of going through this data. I'm just going to show you a subset of very preliminary data right now. This is data that I've matched with life history, so body mass and lifespan of the individuals. I'm looking at 138 species. Before we had 36 mammals, now we have 77 species of mammals. We're now branching out to look at outside of mammals, so we have 20 species of reptile, 39 species of birds, and sorry, poor amphibians, they're not very represented, at least in my subset I'm looking at right now. We do have more data, but again, this is very preliminary. So here's where the caution comes in. Right now, all of this data I'm looking at is from a pathological service that codes it as abnormal proliferation. We'd like to get better at detecting if it's actually a cancer or a malignant cancer, but right now we're just looking at abnormal proliferation. So that's a big caveat in this data. And again, it's zoological data, and we're looking at abnormal proliferation right now. That being said, about 18% of these 84,000 individuals had some kind of abnormal proliferation. We're calling it neoplasia prevalence. We're really excited because we have a wide range of life history variables just in our small subset of data that we're preliminary looking at. So lifespan, the shortest being the bee eater, that's 2 years, to the African elephant that can live a maximum of 80 years in captivity, to the green anole, that's super tiny, 4 grams, to, again, the African elephant is the largest and most long-lived animal in the current data set I'm showing you. And similar to the plot you saw earlier, we're having percent of neoplasia here times body mass times lifespan. Over here, the color is either red for amphibian, green for bird, blue for mammal, purple for reptile, and the size of the dot is the number of individuals in the data set. And like I said, all animals, we saw an 18% prevalence of abnormal proliferation. In amphibians, we saw 4% of them. Birds, there were 10%. Mammals, 26%. And then reptiles, 9.5% of them had some kind of abnormal proliferation. This previous plot I showed was just mammals. Now we've included a much bigger expansion. The poor mouse lemur is up here with the bush baby. We've rechecked this data a bunch. We're going to keep rechecking it. Super high rates of, we can talk about this in the discussion. But also the ferret is pretty high. There's over like 10,000 ferrets in our data set. And even when I was talking to the vets when I first started this project, they called out the ferrets as number one, being an animal that they just know anecdotally as an organism that gets lots of cancer. Okay, and then alligators are just amazing. They had the lowest prevalence of having neoplasia in our current data set. Previously, I showed that there was no relationship between body mass and lifespan and cancer rates. Now I am showing you that our data supports that there's a negative relationship between body mass and lifespan and cancer rates of the current data. So bigger, longer-lived animals do get less cancer from this data set. And we can take a subset and look at primates specifically and go back to, this messed up a little bit. In an attempt to do this quickly and add human, it didn't show the human rate. So earlier I told you it was 1 in 4 men and 1 in 3 women would have a lifetime risk of cancer. So visualize this human coming back up here. And what's kind of surprising to me is they're grouping right along with a similar rate as to other great apes. So maybe we're actually not that different in our cancer rates, with the caveat that, again, these are primates living in zoos, okay? These are not wild primates, okay? I also want to go back to the point that mammals seem to be getting the highest rates of neoplasia in our data set. And this is a question that's starting to really pique my interest. Is there a sampling bias? Do zoos love mammals maybe more than other organisms in the zoo, and they're getting necropsied? Necropsies typically cost a lot of money, and so maybe zoos only do it for bigger species. It doesn't seem to be the case, but always it could be. There's also a good representative of birds in our data set that have necropsies, and they just don't get as much cancer as mammals. Could be something with the environment or our closures. But I really want to focus on maybe it's evolutionary pressures. Maybe there's something else going on here that makes mammals more susceptible to cancer than birds and reptiles and amphibians. And there is some ideas out there in the literature that it might be due to that we're placental mammals. And we have internal gestation, and people have put forth the idea that the placenta is very cancer-like in its phenotypes. There's selection for invasion and angiogenesis, that's getting blood vessels into to feed the fetus, and also immune avoidance of the maternal immune system. And for those of you not familiar, there's different placental invasion types across mammals, going from least invasive to most invasive. This is the fetal tissue, this is the maternal tissue. Here you have an interaction where there's just very little invasiveness, to hemocorial placentas. They're the most invasive placenta type. They actually break through the mother endometrium lining. They take these maternal spiral arteries, actually remodel them, and get direct access to resources and blood flow for the fetus. And humans do have the most invasive placenta type, the hemocorial placental. So with this idea, there's people that have predicted that there's a link between placenta type going from least to most invasive, and mammals with a hemocorial placenta have selection for an invasive cellular type, which could be really bad in getting cancer later on in life. You have selection for genes for invasion, and those can be turned on if the cell is now not cooperating and following the rules of the multicellular body. There was previously a study that showed that cancer malignancy covaried with depth of placentation, although this study only looked at epithelial and then endothelial placentas. In our study, this is a subset. I've done it for a larger subset of placenta types or animals as well, but we actually do not see a relationship between placenta invasiveness and malignancy. We don't see it for just occurrence of cancer. Any way I look at the data, there's actually no relationship between placental type and cancer rates, which I was surprised and sad about because I really liked the placenta story. But what else could it be? Maybe it's just a binary function. If you're a mammal or not a mammal, then maybe you're more susceptible to cancer. Maybe placental mammals are not as good at detecting cells that are not our own. During internal gestation, I'm just going to briefly talk about this. During internal gestation, there's a transfer of cells from the mother and the baby back and forth. And we know now that these cells stay within an individual for a lifetime. If you're a placental mammal, you're chimeric with this swapping of cells. And at least within humans, there's great evidence to show that your mother cells stay within the baby for the rest of their lifetime, at least into the adulthood. And the mother has fetal cells floating around in her after pregnancy for the rest of her lifetime as well. And so this is quite shocking when if you were to take an Immunology 101 class, you learn about self and non-self, and everything that's non-self gets cleared from the immune system. And that this is not necessarily true for placental mammals. And so maybe this is a consequence of placentation where we're not so good at recognizing self and non-self, because we have to live at least with a paternal genome in us for a few months while we gestate, and then this byproduct of cells that slough off into each other afterwards. And this terminology of these cells transferring back and forth is called microchimerism. We can talk about it more in the discussion. I have some fun ideas about what these cells are doing after a mother gives birth. Okay, so going back to cancer rates. How are we on time? Okay, we're good. Across life, what we see is that there is, again, a variability in cancer rates across organisms. Even within mammals, there seems to be some mammals that are very good at defending against cancer. And so then you start asking the question, why species vary. I put up the really adorable naked mole rat as the poster child for cancer, anti-cancer organism. They've been studied for years and years and years. Only a few cases of cancer ever. There's a really great group led by Vera Gorbanova, and she's figured out some of the molecular mechanisms. They're quite fascinating. They're also the only eusocial mammal. They have a queen and a colony. But again, why do we have the naked mole rat here that's seemingly cancer resistant? And the Virginia opossum, which is just as adorable, seems to get a lot more cancer. And And you can start asking, how has evolution solved these different problems of cancer? What are the molecular mechanisms underlying this variability we see across the tree of life in cancer defense mechanisms? And it's kind of like running simulations. Evolution is running simulations for us because it's solved problems across the tree of life differently for different organisms. And we can start mapping these and figuring out what's better at treating the human condition. Okay, apologies for how small this is. I'm going to focus on, this is what we currently know about cancer defense mechanisms across mammals. We have the elephant, who we recently discovered has an expansion of a really important tumor suppressor gene called P53. Humans have one gene, 2 alleles for it. Elephants have 40. I'll show you the functional work in a second, but the field got very excited about this, and then everyone started looking through the whale genome, looking for expansions of P53, and they did not find it. Whales have solved the cancer defense mechanism a different way. They have different molecular mechanism. Along with that, interestingly, the blind mole rat, the cousin of the naked mole rat, is also somewhat cancer resistant. And they have a common mutation in this gene P53 that elephants actually have an expansion of. This mutation is common in human tumors. But this is just a variation, a SNP in the blind mole rat population, but they're seemingly cancer resistant. So there's a bigger picture story, and it's really exciting to start tease apart and figuring out how animals have solved these cancer defense mechanisms. So previously I said elephants have duplications in P53. That was genomic work. We then, collaborators of mine met up with a pediatric oncologist, Josh Schiffman, and he was studying a condition called Li-Fraumeni syndrome. This is a genetic condition in human populations, and individuals with Li-Fraumeni syndrome only have one functional P53 allele, okay? They have a genetic mutation in the other one. The lifetime risk of getting cancer is about 90%. And so when he heard about that elephants have 40 extra copies, he said, oh, I have these cellular assays I want to do with elephant cells. And so this is looking at the apoptotic rate, so cell death, in different cell lines. So cell lines of individuals with Li-Fraumeni syndrome that have one copy of P53, healthy human controls, 2 copies of P53, and then elephant lymphocytes, so 40 extra copies. They challenge these cells to DNA damage, and then they see what happens. So how do the cells deal with the DNA damage? And the elephant cells apoptosed more often than the human cells, all right? And in human cells, apoptosed more often than the individuals with Li-Fraumeni syndrome. And so if you're looking at cell with mutation, elephant cells are doing this more often. They're not taking these risks, right? It's a pretty good cancer defense mechanism to just kill the cell if there's something wrong with it, then try and repair it or live with the damage. And I think this is really exciting because we're looking at organismal level phenotypes and seeing the cells respond. We say that elephants invest in somatic maintenance over reproduction, and then we're seeing that they actually, in functional assays, invest in somatic maintenance, all right? Another interesting thing that the study looked at is they looked at ages of different elephants from 2 to 69 years old. And they saw that this response declined, this apoptotic response declined over the age of an elephant. So younger elephants were better at responding to this DNA damage than older elephants. Oops. Okay. So going back to looking at primates, I have a grad student who's solely focused on looking at cancer in non-human primates. And we've gathered some cells from the San Diego Frozen Zoo. and are running very similar experiments looking at non-human primate, but we've also thrown the elephant, and we have a collaborator that's interested in looking at the Galapagos tortoise, which is awesome. And so right now we're just looking at number of cells viable. So opposite of what you're seeing, before we're looking at apoptotic rate, so higher meant that more cells were dying. Now we're just looking at how many cells are alive, so they're dying. And again, we're giving them a DNA damaging agent. And what we see, we have gorilla, chimp, orang, a ring-tailed lemur, tortoise, elephant, and then human. And what we're seeing as you increase the concentration of the DNA damaging agent, cells start to die. The elephant and the tortoise cells are more sensitive to the DNA damaging agent and going through apoptosis. We think it's apoptosis. It could be cell cycle control arrest. But the great apes are lumping together as having a somewhat similar response to this DNA damaging agent. This is one individual per species. We obviously have to follow this up a bunch and look at functional assays. But I think it's pretty interesting to show that these cellular phenotypes are mapping onto what we were predicting would happen. Okay, so for the last few minutes, I'm going to talk about how this relates to looking at cancer variability within humans. And ask the question, can life history theory help us explain some of the human variability across cancer rates? Where I showed you there's individuals or organisms that invest more in somatic maintenance than others, and we see how this maps out with different cancer rates across the tree of life. And so just how organisms are influenced by environmental cues, resource predictability, extrinsic mortality, maybe individuals could be calibrated early in life to a life history strategy. We know that pubertal timing can be accelerated in stressful environments. And so adding to that framework, can somatic maintenance also be shifted? And in stressful environments, is there an individual that's set up with less investment in somatic maintenance? And you'll see cancer outcomes more frequently in that population, all right? So a prediction just strictly from life history theory, individuals in unstable, unpredictable early environments may have lower cancer defense mechanisms. And I think this is a very useful framework, and it's a really important thing to study right now. We currently don't have very good predictors of what individual is going to go on to get cancer. We can show that there's certain lifestyle risks, such as smoking, that can increase an individual's likelihood of getting cancer. But we know that early detection is critical, and we don't have a lot of good early detection markers yet. And so figuring out what's going on early in life, how it can influence an individual later in life, I think is going to become a really important key into how we can tackle treatment options for individuals, and at least screening options for individuals that might be at a higher risk. Okay, that being said, we can think about it very similar going back to this, where individuals in environments with unpredictability that maybe have to grow quickly or invest in something else might not take the time to repair the DNA, or they don't have the certain genes turned on. You can look at methylation or expression of tumor suppressor genes, and maybe it's costly to actually repair the DNA if you have to fight infection or invest in reproduction really quickly. There's some literature support, although it's very little, looking at early environments and cancer risk. There's the Adverse Childhood Experiences study, and one of the leading causes of death in adults were cancer. There was a study that looked at childhood adversity and increased risk of lung cancer, and is only partially explained by smoking, suggesting another possible mechanism, and a significant association between childhood physical abuse and cancer, and again, lacking a mechanism of what's really going on here. I just wanted to highlight that we're trying to spearhead looking at this early life adversity and just a subtype of cancer, breast cancer, in the Utah population database in collaboration with Ken Smith. And I love using this photo of him because he was explaining how amazing the Utah population database was to Joe Biden when he came by to look at the cancer moonshot and give funding. Okay, so what is the Utah population database? It's an extensive demographic and medical database. It's got 7.3 million individuals in it. And so I'm looking at breast cancer. There's an important distinction between breast cancer. There's two subtypes. There's estrogen receptor positive, hormone positive, and hormone negative. Hormone positive is the more prevalent cancer type. But estrogen receptor negative typically has poor diagnosis and worse outcomes for patients. Estrogen receptor positive breast cancer we're getting increasing evidence that it's linked to reproductive factors of our modern Western society. So things like nulliparity increases the risk, reduced lactation, longer inner birth intervals, anything that increases a woman's cycling, so higher levels of estrogen in her body, has been associated with an increased risk for estrogen receptor positive breast cancer. But these fall apart with estrogen receptor negative breast cancer. And so in this subset, I want to look at reproductive factors, but also predict that individuals potentially with early life adversity, you're going to see a different subtype of breast cancer, and it won't be this hormonal one. It will be the non-hormonally driven one. These are just a subset of the variables that we have on the individual. The Utah population database has so much information on individuals, so we can start looking at getting at how the early life of an individual is by looking at parental age, if there's a parental or SIB death, if there's a remarriage, parental SES. We can look at SES at birth, birth weight, density of kin in the community, and we have information on the individual too, looking at her parity, if she had a child death, her age at last birth. And then what we did was we were able to merge the Utah population database with the Utah Cancer Registry, okay? And at the Utah Cancer Registry, we can have age of diagnosis and receptor status and SES at diagnosis. And then we can ask the question, do early life measures influence breast cancer susceptibility? We have 20,000 cases, 120,000 controls, 16,000 women with estrogen receptor positive, 4,000 with estrogen receptor negative breast cancer. I won't go into it. I'm really excited about this study, that we haven't completed this study. I'm just showing the potential of the data. I'm really hopeful that we're going to be able to see some patterns here. But this is just the variables that are coming out of it. We do show that, just briefly, estrogen receptor negative women do have an earlier age of diagnosis than estrogen receptor positive. And also just crudely looking at the data, age of diagnosis increases with the number of children for both estrogen receptor positive and negative women. More children push the age of diagnosis further, which is consistent with the literature right now. So really excited to move forward in this study. And then lastly, I just kind of wanted to go back to this stat that I threw up showing that cancer is obviously a big concern for us as a society. But throw out the question, how do you cure something that we see across the tree of life? It's essential function of being a multicellular being. And there's a really horrible metaphor about winning the war on cancer and trying to cure cancer and how to cure cancer. And I would argue that because it's an evolutionary ancient disease, it's a consequence of somatic evolution, that we should focus on controlling the disease instead of curing it. There's some really exciting therapies out there that have an evolutionary approach to it. And the goal is to actually maintain the tumor population instead of eradicating it. And they're in clinical trials now, and it's looking pretty promising. But to get away from this war on cancer metaphor and thinking of cancer as something that we have to go to war against, and that there is immediate cure in our immediate future, is maybe something we should focus on controlling it instead. So with that being said, I'd like to acknowledge my collaborators. I'm really happy to say that we just got funding for 5 years to keep studying evolutionary approaches to cancer. And then I'm going to do a shameless self-promotion that I am recruiting a postdoc. So if anyone knows anyone, just email me. And yeah, I can take any questions. So thank you.
Really cool presentation.
Thank you.
So one of the things that might increase the costliness of going through apoptosis is not having the raw materials necessary to, or enough of the raw materials necessary to make a new cell. So I was wondering about fatty acids, and since they're important in creating cell membrane, if you had any thoughts on whether or not the availability of that could sort of turn the dial of how much apoptosis?
Yeah, I actually have no idea. That's a really good question. The flip around is, why do elephants seem to have this unlimited resource to be able to kill? It seems like there should be a constraint. That, why do they have more proliferative potential, I guess, to go through apoptosis so much more than human cells have the ability to do? And we don't know. Yeah. They might have more stem cell populations. We're not sure, but yeah, that fatty acid one is a super interesting question. I have no idea. Yeah.
First of all, thank you. This is a super interesting talk. So one thing that sort of one thing that became sort of clear to me, and has, I guess, been clear for a while, is that cancer is not a monolithic phenomenon. Each of these are sort of individual illnesses to be understood in sort of different evolutionary contexts. So this maybe doesn't apply to some of the more, the cancers that are tied to, androgens or estrogens. But one thing I've always wondered about is, to what extent is maintenance, as we talk about it in the context of life history theory, about endogenous factors that are capable of, reading and editing the DNA, as opposed to behavioral factors that can feed back in and, up or down regulate. So, think about skin cancer and use of sunscreen. Like that is, there's a behavioral proclivity for, conscientiousness or whatever it might be that drives you to apply sunscreen more regularly, so that reduces those risks. But there's a lot in there. I guess my question is, how do you think of behavioral maintenance as figuring into these models?
Behavioral maintenance, so you're meaning adding sunscreen, or?
So maintenance within life history theory is, I think, often sort of spoken about in the terms that you presented here, where it's you've got, endogenous factors, but how is behavior factored into maintenance, I guess?
Okay, so looking at, so that one's a little tricky, because there's a lot of lifestyle factors that influence cancer, including smoking and drinking and being out in the sun and not putting on sunscreen. But how costly is it to not do those things as cancer defense mechanisms? Is that your question? So how?
Well, we can talk about it.
Okay.
I have a lot of thoughts on it.
Okay, but am I getting at it, or I'm happy to explore this further?
I think it's a little too involved for
Okay, yeah, sorry. Yeah.
Hi, great talk.
Thank you.
So maybe this is a dumb question, but does trophic level matter? Have you thought about in the cross-organism comparisons,
Mhm.
I'm just thinking that it seems to, like, we think about eating meat and things like that as being upping up cancer risks. I'm just wondering if you looked at trophic level and if that seems that, alligators is a lot of meat.
Yeah, alligators too. But no, we haven't looked at that necessarily. I don't know if that would matter or not. It'd probably be dependent on the organism and the ecology it evolved in, right?
Yeah.
So, but yeah, I don't know. We haven't looked at that. It's interesting though.
So I think now that your data set is expanding so large, potentially you can ask more refined questions than you have been asking because you have better resolution. So size, organismic size is obviously a factor, just for the simple reason that you gave the number of cells. But, and keeping in mind that the rate of proliferation and replacement of tissues differs across tissues, tissues don't scale evenly with size.
Yeah.
So as an animal's size goes up, its total somatic mass goes up, the size of its kidneys goes up proportionately, but the size of its skin proportionate to its total somatic mass diminishes, right? So now skin is replaced relatively quickly, so you have a problem with high proliferation. But you would expect, for example, that, I don't know, maybe subcutaneous fat that isn't replaced at the rate that dermal cells are, is not going to show the same relationship with size, all else being equal, than, say, nephrons are, right?
Yeah, absolutely. We're really excited to look into this with more detail, looking at different types of cancers, and we have that data of where these cancers are in different organisms and start teasing apart. There's a big debate in the cancer community looking across organs and looking at more proliferative organs getting more cancer, and it's a little misleading. There was a paper that was recently published showing that the most proliferative tissues do get more cancer, but their math was a little wonky. I won't go into the details, but there's been a lot of, it's a current debate in the cancer community whether that actually exists or not. But I think we can start teasing apart that. The other exciting thing is I just saw a talk by an individual that's just looking at somatic mutations in normal tissue and getting very shocked at what the results are. So just looking at normal skin tissue and finding the degree of somatic mutations that happen on a normal individual without cancer is almost as high as the mutations you would see in a tumor. And so that's throwing a curveball at the community because we have high mutational somatic mutation burden, but no, the constraints on that individual getting cancer, so far they don't have cancer. And will they go on to get cancer or not? We don't, it's a study that we can start following, but looking at the normal rate of mutation across tissues is going to be a really exciting field too, and if it differs across organisms as tissues get bigger or smaller. Yeah, it's super exciting.
Very interesting talk. I was just wondering if you had any thoughts about childhood cancers or super early life cancers, whether that exists across species, or are these all sort of senescent cancers?
So the age factor is something that we're pulling out of the database. Currently, we don't have age as anything in this, but we are pulling it out. Typically, juvenile cancers within humans and early developing cancers is with a strong genetic heritability. So it's usually an individual that has Leigh-Fromini syndrome, so there's already a heritable mutation. It's not this natural process of accumulation of somatic mutations. That being said, looking across the data we have right now, we do see some infant and juveniles of different animal species that do get tumors as well. It's likely to happen. So yeah, it'll be interesting to start teasing that apart. we're not unique in cancer at all, and probably having genetic mutations that get passed on that end up having our juveniles susceptible to cancer, or other organisms having juveniles susceptible to cancer. Yeah.
Within mammals, you talked about placental mammals, but I was wondering, is there any data on monotremes or marsupials and if they have lower rates?
I had marsupials in there, I don't have monotremes.
Okay.
I would love to get data on monotremes, yeah. I think it's a obviously important outgroup to look at in looking at the placental mammal versus non-placental mammal data point. The marsupials, at first, the patterns, I was kind of, when I was looking at it, I thought, oh, marsupials are getting a lot of cancer, but it's mostly driven, they had a genetic bottleneck, and what you'll see is a lot of these organisms that have high rates of cancer, almost all of them have some kind of genetic bottleneck in the population. And that's what happened to the Tasmanian devils, and that's, now they have these facial tumors, but even before that they were getting a lot of cancer. We're seeing that in the cheetah populations, they have a lot of cancer compared to some other species. So, yeah, what was the?
I guess I was wondering if there was if they had lower rates versus placental mammals because it's not as invasive.
Yeah, we don't know yet, but we'll see. But yeah, the marsupials, we have to take into consideration that there's also this extreme genetic population bottleneck in some of the island animals there. So, yeah.
Any thoughts on why these genetic bottlenecks are related to higher cancer prevalence?
Yeah, I would love to know that answer. So, just accumulation, there's not a lot of genetic variation, so if you got a certain number of deleterious genes, they stay in the population more. The other thing is this lack of MHC diversity. Seems like it might be important, and if you have low MHC diversity, you might not have a very good library for detecting when a cell is different or not, but I don't know. It's something that's churning in the back of my head, but I really don't know the answer. It's something that should be really important to figure out. Yeah.
Is there a higher degree of variance in likelihood of cancer onset among men as opposed to women? Just thinking about sort of the conditions of.
So men get more cancer and die of more cancer than women, but I don't know if there's a higher degree of variance. I'm not sure.
You talk about life history theory,
Yeah.
but the age that you get cancer, or especially that you die of cancer,
Yeah.
makes a big difference, whether
Yes.
it's pre-reproductive or post-reproductive.
Yes.
And we're living longer than we used to live.
Mhm.
Are we? Does it matter if we die of cancer when we're 55?
Right. I know. I love this question, and I think we could answer this with the zoo data. We're starting to do survival curves of the animals at the zoo because animals are living longer too in zoo populations, and so comparing that with their wildlife average lifespan and seeing if they get cancer after their average wildlife span. The data's not there yet, but we're excited to start teasing apart this because this maybe can start give us clues about humans and if, this cancer is just a consequence of after post-reproductive senescence. Yeah.
So the way that you were using body mass throughout is basically as a proxy for risk, just by virtue
Yeah,
of
number of cells.
But animals of the same body mass can differ in metabolic rate.
Yes.
And if it's turnover that
Mhm.
matters, then that should give you better resolution
Mhm.
than just body mass.
Yes.
Are you exploring?
Yeah. It's challenging to get BMR for all the individuals in the data set, but we are definitely looking at BMR. And that's why I was slightly surprised that the lemurs and lorises in the data set actually had high rates of cancer because they're known to have lower rates of BMR. So it was opposite of my predictions. But yeah, we're definitely going to be including that in the analysis. Yeah.
Just a quick follow-up on that. So,
Yeah.
does BMR change in, is it significantly different in zoo populations versus wild
I
ones?
don't know. There's a lot of, and here's the thing, it's difficult to study animals in the zoo because the zoos are very sensitive over their treatment of animals. They really care about their animals and they don't want anyone highlighting things that could be leading to an animal getting sick. And so I don't know that answer. I also don't know, some of the animals in our data set are nocturnal, and are they nocturnal at the zoo, or are they completely flipping their physiology? Are they getting skin cancer because they're not usually out in the sun? And so there's a lot of questions that we don't know the answer to, but we hopefully can start teasing apart. But yeah, I know obviously animals in the zoo are extremely stressed out.
Yeah.
So,
Well, and I imagine just their energy expenditure is going to be much lower, right?
yeah,
So-
and they feed more often. There's lack of exercise. They're a really great comparison to some human populations. So, but we'd like to compare the zoological data that we're getting with wildlife data. We might be able to do it in bats soon, so it'll be a nice comparison of captivity and wildlife, and maybe we can start teasing this apart.
So when you were comparing cancer rates between mammals and other types of animals, and finding much higher rates of cancer in mammals, you went through some different explanations. But one that you didn't address, and I'm just kind of curious about, is whether tumor phenotype differs between mammals and other types of animals, and then whether the individuals doing the necropsies are more familiar with how tumors look in mammals, and whether that could influence.
Very good question. We don't know, and there's a big argument that whale tumors would have to be much bigger to kill a whale. And so, yes, they do get as much cancer as but they're just different. And we don't know that. We don't have anyone studying tumors in different animals. We just got our first cell line of gray parrot squamous cell carcinoma. And so we're going to start looking at tumors across animals as well to see if they look similar or different. But yeah, we don't know. Especially the farther you get out from mammals, they might be very different. And yeah, good question.
Going back to what you're saying was interesting about pregnancy. Because it's in an immunosuppressed state, what does cancer during pregnancy look like? Do you see higher rates of sort of cancerous activity?
It's actually really rare. I don't know if we know if it's like a higher state. I know it's very rare during pregnancy, but if it happens during pregnancy, it can pass on to the fetus. So that's one of those weird contagious cancers in a weird circumstance. And so the mother's cancerous cells can transfer to the fetus and cause cancer within the fetus. But again, very rare. There's only a few instances reported of that. So.
I'm sure you thought of this also, but just in case you didn't.
Yeah.
Another feature of zoo-kept populations, both ones behind bars on the inside and the ones on the outside, is that they're evolutionarily novel-free in parasites. And if there's been, particularly in GI macroparasites, if there's been a co-evolutionary arms race where the helminths, for example, have all these anti-inflammatory secretions in order to induce tolerance in a host, then you would expect that the novel circumstance of their absence is going to result in proliferation that you wouldn't see otherwise, with circumstances for mutation, so inflammation being a dangerous conflagration. So do you, presumably, there are lots of deer gut samples available, right? Because hunters shoot deer, but they don't look at their, they don't cook the guts generally, right? So, deer populations that have endemic helminth parasite loads versus zoo populations that are being dewormed regularly. Conceivably you could you could measure differences in
Yeah.
GI tumors, for example.
That's a really good point. I definitely was thinking of this lack of microbial diversity in zoo animals, but I haven't thought about it in that kind of, because that could be really easily obtainable study to do. I also thought about it in the case of every time I hear about an individual deworming a cat and what the consequences are for that. But domesticated species have a lot more cancer than even captive species. We breed them for cuteness, not for health and longevity. So they have a lot of bad genes.
And they're
going on.
bottlenecks to the extreme, right?
They're bottlenecks to the extreme. Dogs have a 10 times higher rate of cancer than humans, a purebred dog. And so, this is a really interesting point. So Peto's paradox holds across populations, but not within. So the number of cells seems to, or at least the body size, seems to relate to higher cancer rates within a species. So within humans, taller humans have a higher probability of getting cancer. And let me see if you people shake their head and go excited. And you can see that within dogs too. Chihuahuas don't get as much cancer as a Bernese mountain dog. So yeah, it's an interesting dynamic where it's looking at cancer selection across the species, but even within the species, then the axis shifts, and you do see increased rates of cancer.
So with placental mammals, you mentioned that maybe the immune system isn't very good at recognizing self versus non-self, therefore leading to higher rates of cancer. Could there be on the other side of the same coin? Could that lead to higher rates of autoimmune disorders?
Yeah, I wonder. Yeah, we've been trying to explore this balance, and maybe autoimmune diseases and cancer are like two sides of a balance. Or if you're really good at detecting or too hypersensitive to detecting, that leads to autoimmune diseases, where if you're not good enough, then you could be susceptible to cancer, and stabilizing selection means you're in the middle, but those tails, you either have autoimmune disease or cancer. The tricky thing is some autoimmune diseases lead to cancer because of inflammation and cell turnover, and then you just get a higher probability of accumulation of mutations in this inflammatory process. So it's a little more complicated than the nice two sides to a same coin thing, yeah. We've been thinking about that a little bit. And we don't know about autoimmune diseases. I would like to do, hopefully now that I'm friends with the vets, we can look at potentially what autoimmune diseases different animals get as well.
Thank you, a wonderful talk. I wonder if you could say a little more about that data set of the people from Utah.
Mm-hmm.
Like, for instance, does it include in the sort of life history related cues from people's childhoods, does it include things like residence changes or whether their parents got in trouble with the law, that kind of thing?
Probably not parents getting in trouble with the law, but it will have residence changes because it's a census that they've been mapping for a very long time. And then any health records, so at least you might be able to see if they're in the hospital for something. And sometimes it'll say why they're in the hospital, so if it was like domestic violence or something like that, that might be in the data set. And then obviously it gets better as the years go on. And the other thing, if we push back and look at the data set from individuals that were really early on entered in the data set, we can look at natural fertility populations too, potentially. So it's a really powerful data set. You can actually go on their website and play around with the type of data that they have in it, and then you can submit a request to see if your study or question would be in line with them, and you can get. It takes a while to get access to, but it's not. They have a website and they want people to start using it because they have all this information. And it's continually collecting. It's quite interesting too because I was with the individuals in this data set, I'm like, oh, I really wish we could have samples of these individuals' tumors, if we could get some blood tissue, but I don't know how many. And they're like, oh, well, our population is so used to like signing consent forms. Like, there could be quite a few individuals in your data set that we already have tissue in our biobank. So it's a really, I think, a powerful direction that we might be able to study some really interesting disease and dynamics in with the database, and then also biobanking some of these individuals. So, yeah.
Hi, thanks for an interesting talk. So I was just wondering, I know you had talked about life expectancy and also age and all those sorts of things, and so I just wanted to know if you gave any thought about telomere length and also telomerase and how that might be interacting with somatic maintenance and their life history perspective.
Yeah, good question. Are you talking about within individuals or across the organisms?
I was thinking
So,
both.
okay, so it's interesting, and there's a really beautiful study in rodents looking at telomerase expression, and so mice actually express telomerase in their somatic tissues, all right? And they get a lot of cancer, but they never get selection for cancer, and they mostly die in the wild. But as the organism gets bigger, it shuts down telomerase expression in the somatic tissues. And you think of that as like a cancer suppression defense mechanism where you don't want your somatic cells the ability to reproduce all the time, right? And so then you cap them with telomeres with a certain length, and then as they replenish and replicate until the point where they can't replicate anymore, then they senescence, and then hopefully they're not a cancer risk, right? Because they stopped replicating before they can pass on bad mutations. We don't know how that looks across organisms other than, there's some really beautiful studies in rodents and a few more in other organisms, but looking at elephants or things like that, we don't know the telomerase dynamics yet. Within human populations, people have done studies looking at telomere length and cancer risk, and it seems like really short telomeres and really long telomeres increase your risk for cancer. So it's a little bit of a complicated story, yeah. And those studies don't actually look at how often telomerase can be expressed to replenish the telomere lengths as well. So when they're really short, they get sticky and the DNA can break because they bind to each other, and that increases the risk for cancer. And when they're really long, they replicate more than they should, increasing the mutational burden. So it seems like having the Goldilocks telomere length is a good one to have.
Okay, well, if there are no more questions, we'll end here.
Okay. Yep.